Thank you. Well, good evening, everyone. I have to say, it's great to see a crowd. Compared to the audiences we've had at previous inaugurals since I was appointed, this is fantastic, and a testament, Vass, to I think how highly we all regard you. So thanks for that. So, for those of you who don't know, I'm Charles French Constant. I'm the Pro Vice Chancellor for Medicine and Health Sciences. I took over from Dylan, who's in the audience, in August of last year. I'm afraid to say, I'm also acting as head of the medical school, so I'm afraid you're going to get a double dose of me today, because I'll not only be doing the introduction, but I'll also be doing the vote of thanks and looking after the questions. So, this is our 5th inaugural lecture of the spring term, and it's great to see you all here. We've also got a number of people online, so that's fantastic. And do let us know in the chat. Keep communicating via the chat where you are and what your questions are, and we will work hard to make sure that questions that come in on the chat are answered. So, as I've already said, our speaker for this evening is Professor Vassiliou, or known as Vass to all of us for convenience. And Vass grew up in Cyprus and then came to study medicine at University College London as a Chevening scholar. And then he went on to do a series of stellar house jobs at the professorial unit of UCL, then Cambridge, Addenbrooke's, NNUH, of course, and Royal Papworth. So a very stellar pedigree there. And then he started his research on cardiac imaging, innovative research on the role of myocardial tissue characterization by cardiovascular magnetic resonance for his PhD at the Brompton, and that, of course, is going to be the platform on which the exciting findings he's going to talk about today are based. After he did his PhD and subspecialization in imaging and further clinical training, he was a consultant cardiologist, first at the West Suffolk and the Brompton, and then coming here as a senior clinical lecturer in 2016, where he initiated the research, some of the research that we're going to hear about. But Vass does a lot more than that. He's also the current chair of the Population Sciences and Public Health section and co-chair of the Certification Task Force of the Association of Preventative Cardiology. I did try and remember all that, but I just couldn't manage it. As a board member of the European Council of Heart Valve Disease, and he's also advised the National Institute for Health and Care Excellence on the recent guidelines for heart valve research. But Vass is also passionate about education. Whilst he was training, he completed a master's degree in medical education and won a regional award from Health Education England. And it's really this passion for education that has been such a key feature for me. I share with all of you the strong belief that the clinical academic ecosystem on the Norwich Research Park, NNUH, the university, the institutes, could be fantastic. It is fantastic in places, but we all want it to be greater. And Vass has done an amazing job there in building up the number of NIHR ACFs and CLs, in mentoring them, and generally creating this environment where our youngsters who wish to be clinical academics can thrive. And for that, I'm extremely grateful, and I'm sure you all are as well. So with that, Vass, I'd like to invite you to give your lecture.
Thank you very much, Charles, for the kind words. And thank you very much to everyone who has managed to find the time in the busy schedules to attend in person. Thank you. And the people who are attending online. Thank you very much. So I will be talking about my journey, really, and the title is Beyond the Black and White Pictures. And what I'll talk is my journey. I'll focus a bit more on the research side, the collaboration, the friendships that have formed through the years, the plan B that we all have to have under conditions that can come without us even knowing about them, and then how the journey continues from now on. At the end, there is a wine reception, you'd be pleased to know, and as a cardiologist, I can assure you that it's always quite healthy to have a little bit of wine. If anybody wants to go to extremes tonight, I think that's fine. You'll be pleased to know we have a liver specialist in the audience, so you will be well looked after. As Charles already said, I was born and grew up in Cyprus, which is a lovely small island. And then I did my A-levels back home and then came to UCL to study medicine. And the years at UCL were really eye-opening for me. I came from an island that has a population of 500,000 to a city that has a population of 10 million, and had the ability to interact with lots of people that we've kept to touch even today, and I'm pleased to see quite a few of them watching this online. On completion of the medical degree, I was very fortunate to get the professorial academic job at UCH and the Middlesex Hospital. It still existed back then, and it was a fabulous hospital. And I would like to pay a little bit of tribute to my first mentors at the time, Raymond McAllister, Arun Kingorani, Patrick Valance, and Hugh Montgomery. All of them, what they had in common was that they're clinical scientists, they were academics. And the way they managed their patients reflected the evidence that they had, the gaps of evidence, and what they saw in clinical practice allowed them to formulate new research to be able to answer those questions. Therefore, they instilled in me this curiosity: what can we do next? What can we do better? And how can we go about getting the evidence for what we need? And that stayed with me forever and started making me inspired to follow a clinical academic career. And then why cardiology? So, yes, you can take it from me that cardiology is important, but what I would like to suggest is that other people have shown that cardiology is important. And when Neil Armstrong landed to the moon, he didn't take an X-ray of his hand or of his head or anything like that. What he did was a tracing of his heart. So he did take an electrocardiogram of his heart, which is what we show here, and therefore indicating, heart is important. No matter if you are in London or Norwich or the moon, that's something we all need to look after. And after my London time, I came to Cambridge for the Addenbrooke's medical SHO rotation linked to Papworth, and subsequently joined the cardiology training scheme in Norwich, and I moved from Cambridge to Norwich with some intermediate stops in Peterborough and Ipswich. And then I decided I wanted to do research. So my journey then led back to London once again, but this time slightly more to the west of London at the Royal Brompton Hospital and Imperial College London. And I'm very grateful to Roasters Charity and the NIHR for funding my research. And I'm glad to see quite a few young people in the audience, young academics, aspiring academics before, during, or just after their PhDs. And this is how my research proposal looked at the beginning. It was great. We're going to steam through. And what in the end happens is something like that. So, PhD research is not always what you aim to get. You try your best, but you have to accept that, there will be changes. And if you remember one thing about today, if you're planning to do your PhD, you need to be able to move and take the opportunity that will be provided to you. And my PhD was using cardiac MRI and looking at the myocardial tissue characterization. And this is the first person that got any MRI pictures. This is Professor Tamedian in 1977, and it took 4 hours to just get this one image. And I think you would agree we have moved on quite a lot since then. Yes, again, I can say that MRI is important, but when Royal Mail says MRI is important to the value of 1.60 pounds, I think we can take it for granted that is important. So I wanted to do MRI research because I thought it was important for our patients. And what better place to do it than the Royal Brompton in London? And I would like to share a little bit of the story about the Royal Brompton here. And what most people will see on the image there is a double-decker bus. But there is a story to it. This is a decommissioned double-decker bus. And when it was out of service, it was bought by the Brompton. And what they actually did, they took everything out and they converted it to an MRI. They didn't buy an MRI and put it there. They built the MRI from scratch. So this was the early 90s. And Brompton Hospital was the first hospital in the UK where they actually had a functional cardiac MRI dedicated scanner, which they built in-house. And the beauty of it being on a bus was if you needed to take it for research to other countries, such as Italy for thalassemia, then they would actually drive the bus there. But by the time I had gone to the Brompton, things have moved on a little bit more. So they now had 4 cardiac MRIs, dedicated cardiac MRIs, scanning about 10,000 people a year, which is massive, and even having fancy things like combined angiography and MRI suite to be able to minimize radiation in people. Only 2 in the country at that stage. And I was very fortunate, and I would like to take the time to pay my tribute to my supervisors, Professor Sanju Prasad, who allowed me to study under him for a PhD, and his overall supervision and support during those years subsequently, co-supervised with Professor Dudley Bennell and Dr. Peter Gatehouse. And the one thing that always stays with me from my Brompton time is that not only did we do the work, but we were a big family. And we supported each other through the research, through extracurricular activities, and this is all of us in Barcelona quite a few years before COVID, where we would all go out after the conference and have some dinner. And this was just the cardiovascular MRI group. So you can see we had a lot of, let's say, manpower, bouncing ideas all the time with each other, supporting each other and moving forward. I appreciate that there are also non-doctors in the audience, so I put a cartoon of the heart. So basically, blood comes in, it crosses through the valves and goes out to the main parts and organs of the body to provide oxygen, and that is what the heart does. And I would argue it's quite important. And what we can see here is the left ventricle, the left side of the heart, and the right ventricle, the right side of the heart in a schema. But actually with an MRI, we can depict that very, very nicely. And it doesn't matter whether the patient is short or tall, big or small, we can still get excellent images with an MRI. And to get this one, which allows us to see the heart, it probably takes about 1 minute or less than 1 minute to do. And then if we change the angle of the sequences a bit more, we can see the heart in a different way. So here we have the left ventricle, the left atrium, mitral valve, right ventricle, and right atrium. So using some very simple sequences, we can see the anatomy of the heart, and we can see how well it is functioning. And then it can allow us to see the valves as well, and I'll take a little bit of time talking about valves because they're important. We have the tricuspid valve, the mitral valve, the aortic valve, and the pulmonary valve. And if we look at how the aortic valve looks under an MRI, for example, we can see here that it has 3 cusps, and they are opening and closing very nicely, and blood flows through that, as opposed to when somebody has a narrowing in the aortic valve, something called aortic stenosis, where we can see the valve is struggling to open, and we can see the structures surrounding the heart are quite dilated, indicating that there is a little bit of back pressure. And occasionally, you get the odd image that brings a little bit of happiness and light in the team, and this is what I call Panther-sign. It doesn't exist anywhere. It is not associated with any, adverse prognosis whatsoever, but it just happens to see it, and it's nice. But what the MRI does do, it allows you to correlate very well, and it was the first and only way that you can see the muscle of the heart and characterize it without giving any radiation to the patients. So here on the left, we have a histological sample from a patient that had a heart transplantation. The pink is healthy, the white is the unhealthy scarred myocardium. And we can see that this is very well replicated on the MRI, and these are early images from the 2000. And when it comes to talking about what the muscle of the heart is made up of, it goes into one of 3 patterns. Either it is black, and these mean it is healthy. Either it has areas which are white in the subendocardium, like here, which indicate that the person had a heart attack previously, or you have this relatively new description of scarring in the middle part of the heart, having normal muscle towards the blood and towards the epicardium, something called mid-wall fibrosis or mid-wall scarring. And every individual, if you scan every individual, they will be able to put them in one of these 3 categories. And it is important because of these 2 people. So Professor David Newby and Professor Mark Dweck from the University of Edinburgh. So Professor Newby was the world leader on aortic stenosis and has been doing research on aortic stenosis for the best part of 30 years. And Mark used to be a fellow at Edinburgh and then subsequently at the Royal Brompton. And what Mark did was to follow the patients with aortic stenosis that had an MRI scan at the Brompton, and he noticed that if they have this pattern of mid-wall fibrosis, they actually have an adverse prognosis. And this was very instrumental because for the first time we could find something that would indicate that you can classify patients with aortic stenosis to the ones that will do well and to the ones that will do less well. From the time that Mark was a fellow at the Brompton, he's now a professor in Edinburgh, and we've continued a very close collaboration. And what I was then able to show is that, yes, it's important to know that this mid-wall fibrosis pattern, the mid-wall scarring pattern, has worse prognosis. But we also knew that the systolic pumping action of the heart, in other words, how well the heart is pumping, also affect prognosis. So if we look at this diagram here, a value of 75 in the left ventricular ejection fraction is better than a value of 50, which is better in turn than a value of 25. And what we see correlates very nicely in patients with aortic stenosis is that as your ejection fraction improves, then your mortality goes down, which is important. If, however, we make the patients and divide them into the patients that have this mid-wall scarring, we see that their mortality is much higher. And if we take the patients that do not have any scar, their mortality is much better. But the important thing is this is additive to the ejection fraction. This is additive to the systolic function. So it's not one or the other. Both of them are independently predictive of mortality. And moving from single-center research to multi-center research, we then got together with Mark and John Greenwood from Leeds, the Leicester team, Barts, and Oxford, and we pulled our data on patients with aortic stenosis. And we had just over 600 patients with MRI, myocardial tissue characterization imaging. And again, what we show is that this is not something that one center sees and the other doesn't. All the centers pulled together have an adverse prognosis when you have evidence of mid-wall scar. And therefore, coming back to the patient, all the European and American guidelines still to date indicate that if you have symptoms when you have severe aortic stenosis, notably chest pain, breathlessness, or blacking out, then it's time to have surgery. Sometimes, if you have evidence on the ultrasound of the heart, the echocardiogram, that there is decompensation of the heart, then surgery might be warranted. But what we were able to show is actually if you have this mid-wall scarring of the heart, it indicates that your heart is already coming under strain. And unless something happens to it, to relieve the pressure of the aortic stenosis, then the prognosis will be worse. And I'm delighted that when I was sitting in the group in NICE that brought out the recent guidelines of valvular heart disease in November 2021, this was something that was acknowledged for the first time, and I'm delighted to see the chair of that panel, Dr. Garby, in the audience tonight. And moving on from there, we had the group of the various institutions, so we got together to start two studies, the EVOLVE study led from Edinburgh and the EasyAS study led from Leicester by Professor Twegg and Professor McGann, respectively, looking at whether we can provide randomized evidence that earlier intervention in these patients can be life-saving. And the study started before COVID. COVID delayed them a bit, but they're now continuing at an acceptable rate. But my talk was not just about black and white picture, therefore I've only been talking about black and white pictures so far, so where's the color? And I will stand to this slide for a bit, which is quite important. And we have the MRI image or the MRI picture on the left, and on the right we have histological assessment from patients who underwent aortic valve replacement. And on the left image, if you see any white, like the areas here, then this is bad, indicating that the muscle has died and it's scarred. If you look on the biopsies on the right, if you see lots of red, like here or here, it indicates that there is scarring of the muscle of the heart. And therefore, if we look at the first, the third, and the fourth panels here, there is good correlation between the MRI and the histology. However, the interesting one is the second panel. The MRI looks normal. There is absolutely no white parts of the magnet, of the myocardium, so we would have called that healthy myocardium. But on histology, we pick a few bits of red, which indicate that there is a fibrotic process, the scarring process is there. And what we're missing is a different type of scarring, something called interstitial fibrosis. So if we look here, on the left, we have an electron microscopy picture from a normal, healthy volunteer. If we look on the right instead, we have a picture from a patient with aortic stenosis. And what we see is this distance here has increased, marked in red, and that is the extracellular volume that has shown expansion. And this is where interstitial fibrosis, a finer form of scarring, takes place. And at that time, we were able to have a new novel sequence, the modified Luke-Locker inversion recovery sequence, or more commonly known as T1 mapping, that was suggesting that it could measure interstitial fibrosis. And this is where the colors come in, because this T1 mapping sequence could come in various colors. So we have purple and blue, and we have red and green, and you could actually post-process this to whatever colors you wanted. But the important thing to show here is that on the left, we have a normal individual. On the right, on the middle, where we see a little bit of red, we have an individual with moderate aortic stenosis, and on the far right, we have an individual with lots of red and a lot of aortic stenosis, indicating that we have no fibrosis whatsoever here. We have slight interstitial fibrosis or scarring in the middle, and we have quite a lot more interstitial fibrosis or scarring on the right. And then having a sequence, it was important to establish whether it was accurate, whether it was precise, whether it was reproducible, and whether it was suitable for the long-term use. And the most important thing here was to appreciate that, yes, we have something, but unless we test it out and we know how it behaves under certain conditions, it might not actually be useful for clinical use. And then we come down to how do we know it's precise and accurate. And the best description I could give is if you try to hit the bullseye and you have 5 darts, if they all go in the bullseye, then you're both precise and accurate. If you miss the bullseye, but all of the darts land together, it means that you're precise, but you are inaccurate. Now, in the MRI language, that is not too bad, because you can put a corrective algorithm in and know exactly what you're doing. But if you're not precise at all, then this is not good news, because you wouldn't be able to use any MRI sequences for clinical practice. And here I was delighted to work with 2 cardiovascular surgeons at the Royal Brompton, and we have continued that collaboration since, Mr. George Asimakopoulos and Mr. Tony De Souza, who would provide lots of samples of patients undergoing aortic valve replacement and other surgical interventions. And also, I was able to work and collaborate with Dr. Katarina Vassiliou from Berlin originally, who is starting a job at the Brompton in the next few months, which is really fantastic. And she was able to undertake quantified histological examination of our samples, and therefore we could correlate them with the MRI. And thinking about the darts, and with that in mind, I thought, yeah, fine. If we throw 1 dart, we might hit the bullseye, but then we could be well off. Whereas if we throw 5 darts and we average them out, we are more likely to sort of converge towards the true value of what we are having. And I thought, why should we just do the convention of just taking 1 slice on the MRI? Let's try and add some more and see what happens. After all, this is research. You know, we don't have the NHS time constraints. We can take the time and understand the sequences a bit better. So I devised these 6 models whereby we had incremental amount of imaging in each one of them. And what we showed is that the more slices you could put in, the more imaging you could do, the better reproducibility you had, and importantly, the better accuracy you had when it was compared with histology. And this was the first report that was published in the same journal that the Molly sequence had been published, which was very nice. And we showed that, yes, we have a sequence that we can optimize, and it's actually, we've reached it to a position that is reproducible and accurate. But then, we know in the MRI world that what works today might not work in a year's time. We know that temperature might affect it. We know that patient characteristic might affect it. And therefore, how did the sequence that we have relate to all of this? And this is when a very basic science component of my research came about, and was actually to join the biochemistry lab together with Peter Gatehouse, who was my co-supervisor, to try and find recipes that would mimic the MRI values for T1 mapping and T2 mapping, and create something called phantom. So this is, when you talk in the MRI world about phantom, it is something like this. Basically, you have it, you put it in the magnet, you scan it, and if it gives you the values you are expecting to see, it means your sequence is fine. And then if you scan it longitudinally, you are able to detect small drifts of the MRI sequence. And we could put it in dry ice, bring the temperature really down, and then gradually lift it up again to see how the sequence of the MRI behaved. And this was quite exciting part. I'm not sure how they let us do this in biochemistry and gave us a free range to use all their equipment, but you'll be pleased to know we didn't break anything. And what we could see is that if you followed this for 12 months initially and subsequently for 36 months, it behaved quite well. And we could see that the various components of the sequence here had a different degree of accuracy. We could see that 3 of them are very stable, but 1 of them was drifting a bit. But actually, if we look at the temperature of the magnet at that time, we could see that it was drifting in the same direction. And when you actually take temperature into consideration, that drift goes away. And what it meant is that if your temperature is changing, one of the parameters of a sequence is not as accurate. Therefore, if you were to scan a patient who had temperature of 40 degrees, let's say, or 39, then you will need to correct that to know exactly what the values were. And likewise, if you have patients who were at the extremes of heart rate, if they were very tachycardic or very bradycardic, then again, you would need to make adjustments to make sure that the values you're getting are as accurate as you want to be. And this led to the first paper indicating the recipe of how to prepare these phantoms, which was subsequently taken by multinational studies as the gold standard recipe for creating the phantoms. And yes, we've now got to the point where we had the sequence, we optimized it, it was working, it's accurate, reproducible, but it does come down to the clinical question, does it actually add anything? You know, yeah, it was good fun, but does it have a clinical implication? And this is work led by Edinburgh and Russell Everett working for Mark Dweck and David Newby. And the way to think about it is we have this middle line. If you are towards the right, it has worse prognosis. Towards the left, it has better prognosis. If it crosses the line, it means that you don't know what's happening. So the ECV represents the colorful maps. The LG for late gadolinium enhancement represents the black and white maps. And therefore, we can see when you put the two together, the colorful maps come as significant and more predictive. But MRI are expensive, you know, it takes time. Not everybody can have an MRI, and it's actually quite an expensive test. So what I wanted to know is, can we get the same information in a slightly cheaper way? And that's when biomarkers came into the consideration. So I was taking blood samples from all the patients and storing them in the Brompton Biobank. And we published quite a few papers showing associations, you know, this biomarker associates with scar, this biomarker associates with prognosis. But these were individual biomarkers. And yes, they're good academically, but they don't actually inform clinical practice. The next step, however, was a big multi-proteomic approach where we actually analyzed about 200 biomarkers that could have some potential association with outcome in aortic stenosis. And using some very powerful statistical modeling, we could now detect the risk of mortality at 1 year and the risk of mortality at 5 years. And just a graphical illustration, you have certain values for these six variables. If they change even a little bit, you can see that you are predicted mortality both at 1 year and 5 year changes. And this indicates that using lots of biomarkers, potentially over and above the patient phenotype, you can actually have a good association with how well that patient is going to do in the longer term. It could, in the future, make you guide the practice and look after the patients in a different way. And here I want to stop and pay a little bit of a tribute to the statisticians that have helped me through this journey. So we have Menelaos from UCL and Simon from the London School of Hygiene and Tropical Medicine that helped me quite a lot during the Brompton years. Aris, who used to be here in Norwich, but then moved to AstraZeneca, helped me subsequently, and Alan, who is one of our senior lecturers here in Norwich now. And I valued very much the discussions I had with them, them understanding what I was after, me understanding how we could achieve it to make sure that what we got was, you know, clinically correct and sound. And we had simple discussions like what is normal distribution, And we also had some more exciting discussions. What is paranormal distribution? And then we started going from an individual parameter on an MRI, an individual parameter on the patient, to starting blending all of them together in a statistically sound way and coming up with risk scores, which we validated to show various things. Simple things like, if you don't have an MRI, but you have some blood tests, can you predict if these patients will have mid-wall fibrosis? And the answer is yes. And more importantly, a paper that was published a couple of months ago, if you have patients that have undergone aortic valve replacement, can you actually predict who is going to do less well in the longer term? And we found that age, quite expectedly, is important, use of aspirin, the scarring on the muscle of the heart. But importantly, we also show that if you have stroke volume as opposed to ejection fraction as a measure of the systolic function of the heart was more important. And for the first time, we showed that if you have left atrial ejection fraction was again an important independent predictor of survival. These indicate that the muscle of the heart is decompensating, but they're not currently used in clinical practice. And I have no doubt that over the years to come, they will take preference over the left ventricular ejection fraction. But I would like to come back to echocardiography a bit, and particularly advanced echocardiography. Some people say this is sort of poor man's MRI. It could be, but if you don't have access to the costly MRI, this is a very good alternative. And some work that we did with Liam Ring from West Suffolk and Papworth after I had come to Norwich. And what we did was to identify patients with mitral regurgitation and look at the left atrial strain as a measure of left atrial ejection fraction. And what we showed is that you could actually distinguish the people that would subsequently require mitral valve intervention. And the process is the same independently of whether you have patients with mitral valve or anything else. And then moving onwards, this is something that we then established together with Peter Pugh's team in Addenbrooke's and with Anna Chusu, who was one of our PhD fellows here. We started looking at the probability of identifying atrial fibrillation in patients who had an embolic stroke. And this is important because if you are able to tease out the patients that will have underlying atrial fibrillation, you will manage them differently and reduce the risk of a subsequent stroke. And again, all this relies on doing advanced echocardiography with left atrial strain. Coming back to the MRI, just to show that the MRI techniques over the years have evolved. So we have here a short axis stack that allows you to get the gold standard function on the MRI, and we can build 3D models with colors of how that looks. We can look at the deformation of the MRI using longitudinal strain or fissure tracking, and therefore during the years the MRI improves a bit more. And I'll come back to the MRI in a little bit, but now I would like to focus on the other side of the research that I've been doing in Norwich, predominantly working with James Smith and his lab. And what we've been trying to do is actually mirror what we see in clinical practice with things in the basic science. And James has been using pluripotent stem cells. And here we have a picture of a stem cell that is hypertrophied, a picture of a stem cell that has scar, and here we can see a stem cell that is moving, and we can actually assess it and see how well it is doing metabolically. What's the energy state? Is it moving in a fashion that is efficient, or is it moving in a fashion that is not efficient? And the important thing about this is we can actually mirror each one of the steps with an MRI. So we can see the hypertrophy, we can see the scarring, and these are the nice color maps. But extremely importantly, and very privileged that we can do this in Norwich, we are able to do MR spectroscopy. And MR spectroscopy allows you to identify how well the energetics of the heart are doing. And Donny Cameron, who is a lecturer in medical physics, has been instrumental in getting this going and validating the sequences. And we are only one of 6 centers in the United Kingdom that are able to do this MR spectroscopy, which is really nice. And then recently we've had Pankaj Garg, who is an expert of 4D flow, and therefore we can, to put it plainly, track the red blood cells inside the muscle of the heart and identify any early features that would indicate that something might not be going quite right or in the right direction. And this is Starry Nights by Van Gogh, and it's an immersive experience. You know, you can go in and immerse. And I wanted to put this here because what is science and what is art? Do they blend together? And when can we be sure that that will help science? And coming back to the question is, again, what is science and what is art, and how do we distinguish the two? And the path, at least until before COVID, was to identify a problem, get a therapeutic intervention, get some basic science support to corroborate the clinical findings, do a clinical translation, do a study, and base some biomarkers and an endpoint on MRI and see how that change in practice could influence things. And of course, as academics, we all need money. And we keep on asking Sanda for a research grant. And some people say, you should stop believing in research grants. And then 2 years ago, this little fellow came, didn't ask, knocked on the door and got inside. And this is the SARS-CoV-2, which causes COVID. And basically, the way it enters the body is it locks onto angiotensin converting enzyme 2 receptor, ACE2. And what that does, it allows it to be sucked in the body, replicate, and then infect the cell and kill it. And the ACE2 levels are quite high in the lungs, the bowel, the heart, and also liver, kidneys, and arteries and veins. And these were the organs primarily affected by COVID when it first started, and we didn't know why. And the reason I mention this is early on in the process of COVID, there was a theory that if you take blood pressure medications or medication for heart failure, notably angiotensin converting enzyme inhibitors or angiotensin receptor blockers, these would be bad for your health because they would allow COVID to get into the body more easily, and therefore your outcome would be worse. And indeed, the first reports of mortality from COVID were in very multi-morbid patients taking ACE inhibitors. And it was taken up by the media, and it had an adverse effect on our patients because they were unilaterally stopping the tablets or phoning in, should we stop the tablets? I had friends asking me what they should do about themselves or their loved ones. And bottom line, this led to about 20% reduction in getting the medication during COVID, and I'm sure a lot more collected the medication but didn't take them in the UK. And we didn't know, we didn't have any information. And at that time, the university was closed, all the labs were closed, research at the hospital had been suspended. So it was a time that we could do get the team together and do some work on deep systematic review and meta-analysis of the information that was available. And Ranu Baral had done a master with me previously and was an academic foundation doctor at the time. And Maddy was the student doing a master with me at that time. So we got together, did an analysis on do the ACE inhibitors and the angiotensin receptor blockers actually affect COVID outcomes in any way. And if we separate them, patients that have hypertension, we see that actually no, it's on the left of the line, it could potentially be protective. If you were taking these medications for any other conditions such as diabetes or renal failure or heart failure, at the very least, it was not having any worse outcome, it was neutral. And excuse the pun, but this did go viral. So I had multiple interviews with the BBC, multiple interviews outside the UK, and the one I've selected here is from Brazil, because at the time Brazil was going through their big first COVID wave. For those in science, we had an altmetric score that indicates it was in the 0.01% of all the articles ever published, all the major newspapers. And what we managed to do at this time was to actually reverse the public opinion that these medications are potentially bad, and emphasize that you should, if you have been prescribed this medication, you should continue taking them, because the underlying reason that you were prescribed the medication in the first place is what will kill you if you get COVID. And then lots of people were asking, should we just take them prophylactically? And there is no evidence for that. And this was the first time that I started using social media as well. Everybody was locked in at the time. People were not talking to each other. And I thought we really need to get this message out that if you know people taking this medication, please ask them not to stop them and continue. And a lot of my friends said to me, do you do any other research, or do you just do research on COVID because you don't post anything else? And I thought, yeah, I do a lot of other research. I spent all my years doing other research. COVID is something that came up in the last couple of months, and we could do this work over a few weekends in our free time. And those of you that are aware of Big Bang Theory, you will understand what is coming. But basically, it's a group of physicists in Caltech in California, which is #1 institution in the USA. And they're talking to Professor Brompton, who is a scientist himself, but he was a TV star and had a program for science for children. And this is effectively a short clip I want to play.
You know, I'm a real scientist. I have a PhD from Cornell University.
Yeah, it's great. Did you bring your pup?
So did you bring your puppet? And it felt something similar, you know. Do you do any other work, or is it just COVID? So moving on, we just continued on COVID because we couldn't actually do anything else at that time. And then we expanded our work and now included more than 100,000 people, allowing us to go into a very deep granular level of what is actually causing the deaths in COVID. And what we could show, it was not the ACE inhibitors or the angiotensin receptor blockers that were causing it. In fact, they would be beneficial if taken, and subsequent randomized studies confirmed that. But actually, it was the comorbidity that was causing the problem. So if you had diabetes, renal failure, COPD, those were the things that make patients have an adverse outcome in COVID. And if you could improve on those outcomes, obesity, for example, hypertension, if you could improve on those outcomes before you get COVID, that would be beneficial. And certainly, if you're taking this medication, keep on taking them. And I would like to take a second to pay a tribute to all the healthcare workers in the UK and abroad, and particularly our NHS for doing as much as anybody could do, and earn huge respect of all the superheroes as shown in this image. Now, moving forward, the future is to sort of understand the mechanism of the pathology, do the clinical translation, see what is important, get the therapeutic target, and then test it in a randomized study. We need to widen patient participation, and that's why Norfolk is quite appealing for that. Use advanced imaging, MRI, echo, whatever is available, and build a network of collaborations. And certainly, locally, collaborations are very easy because we're all within walking distance from each other. These are all before COVID, and it was really nice to be able to travel and see people. And I'll just pick a few people from here that I want to mention. Claire Raphael, we share the same office at the Brompton. She's now an associate professor in Cleveland. Merlin has moved on to Utah. LA is a consultant at St. Thomas's, and Ealing is a consultant at the Brompton. We have here Mamas, who I met in a conference over a discussion about whether to use Twitter or not, and I was not a big fan at the time, but he changed my opinion. Luckily, we started collaborating, and we had a quite seminal paper on cardiac treatment of patients with cancer that is changing practice. Ranu was an academic foundation doctor and done an MSc with us here, and Brody was my previous PhD student. And I was very delighted. They went to the ASC, presented their poster, and got the best poster award of the whole conference, which was nice. Gary was another student that worked with me at the Brompton, who is now a reader himself down in Kent, and then some of the colleagues from Cambridge and Basildon, and Cambridge again, Christie, and Anna, who was one of our PhD students previously. I'm not going to read all the names out, but lots of people to say thank you. These are the PIs at the Brompton, the fellows at the Brompton, the medical students at the Brompton, and then the collaborations that led from that. Mark and Dave have spoken about. Andrew Jabbour was a fellow at the Brompton, now moved to Melbourne, and we've subsequently done a randomized study using this T1 mapping, this tissue characterizations on patients following cardiac transplantation, and John Greenwood, who is the president of the British Cardiac Society, and A.L.M. Levitt from Leeds, where we've undertaken some deep phenotyping work in patients with long COVID. I will take a little bit of time to thank people here in Norwich. Helen, who is my line manager, and I couldn't ask for anything better. James, we are working quite extensively with James and Johannes in the basic science work. Pankaj and Marcus from cardiology. Simon from the interventional cardiology side at the hospital, and Michael, we did some work on dementia and atrial fibrillation, and Donny, who is a medical physicist, optimizing our sequences and making sure they all behave. And of course, I would like to thank Dylan, Michael, and Alistair, who were the people that actually brought me to Norwich in the first place. all of them happily retired since. I would like to thank the cardiology team at the hospital, a very friendly team that gets a lot of good work done, trying to look after patients as well as possible. Regionally, Anna, after she completed her PhD here, became a consultant in Addenbrooke's. James Rudd is a professor in Addenbrooke's. Liam Bring is a consultant in West Suffolk. And Peter Pugh is one of the consultants in Addenbrooke's. And we do a lot of work focusing at the moment on the role of atrial fibrillation and predicting atrial fibrillation in the future. You know, the last sort of joke of the talk is a wise doctor once wrote, and this is what they wrote. And my question to the audience, which will be answered during the wine reception, is, who was that wise doctor? Was it Anna giving an invited talk here at the ESC Preventive Cardiology meeting? Was it James? Was it Liam? Or was it Peter? I would like to thank the funders that have allowed my work to continue. And more importantly, I would like to thank the students and the fellows that have worked with me. So this was a lab meeting on Zoom during the lockdowns, and we have here Vicky, Johannes, and Ioannis, all cardiology registrars at the time, went back and supported the service. Omar was a medical student. Tash was an ACF at the time and now doing an MD with us. Efthimia was a master student. Starusha, a clinical research fellow. Ranu was a foundation doctor. Anna was a PhD student at the time. Rahul, an ACF, and Marcius, an ACF. I would like to thank Matti, Nick, and Brody, who finished their PhDs and moved on with their careers. And then the new people that have joined: Isabella, Isabel, Thin, Kieran, and Hosam. So thank you very much for joining, both in person and virtually. I think the wine would have got room temperature by now, so it would be a good time to finish this. Thank you very much.
Thank you, Vass. I think before the wine, we might interrogate you a little bit. So that was wonderful. Thank you. I mean, listening to you talking about setting up your PhD, it really made me think of a book that I found very helpful when I was starting, by Peter Medawar, who was a famous immunologist. And he wrote in this book, which was called Advice to a Young Scientist. Have any of you read it? It's a fantastic book. Anyway, in it, he basically said, when you're actually deciding what to do, make sure it's both interesting and important. Because his point was that all science is interesting by its very nature. Any problem is interesting. The example he used was spots on a sea urchin. But he says you've got to make sure you actually do something important. And that's what strikes me about what you've done, Vass, both in your PhD with this fantastic work on aortic stenosis and those beautiful, beautiful images, and subsequently, even through the pandemic, you've continued to do really important research. So thanks very much.
Thank you.
Okay, questions, anyone? Can you just get the microphone so that people can hear?
I suppose sitting here as a hepatologist, it sort of feels like you're talking about my organ for many of the things that you correlated. But I suppose my question is, the reason for we see a difference in fibrosis driven by genetics. Do you think there's a genetic component that's driven there, like the DCCK1 gene or something like that? And my second question is, we know from T1 mapping in the liver, where we use it as a correlate of fibrosis, often it relates to inflammation. And with the serous red staining, and I presume proportionate collagen staining that you've done, do you see lots of inflammatory cells there as well that correlate with that T1 staining, or is it just the fibrosis signal, Vass? And is it anatomically distributed in the wall of the heart? Is there a particular area it likes to occur?
So thank you, Simon. Yeah, I wasn't going to point out to who the liver specialist was, but given that he volunteered, if you have a little bit too much in the end, Simon is a place to go. So the first, I'll start with the second question because I remember it. T1 mapping, yes. So T1 mapping, this was a simplified version of what T1 mapping can do. And unlike the liver, which is stationary more or less, the problem we had in cardiology to get the T1 mapping going was because it's moving. It allows you to see both fibrosis and, to a degree, edema. And therefore, if you want to be more scientific, you interpret T1 mapping, which shows fibrosis and edema, in the context of what is called T2 mapping, which is just edema. And therefore, you can deduce of what you see in the TO mapping, how much is edema and how much is fibrosis. So that is T1. Would you mind repeating very...
Why do you think there's differences, Vass, in the different people? We've got the same...
Genetics, yes, genetics. So genetics is very fascinating in cardiology and it's very fascinating in valvular heart disease. And I don't think we have all the answers. And we will get a lot of the answers, mainly due to projects like the 1 million women that started from Oxford and followed up 1 million women. They biobank them and they're waiting for that cohort to get a bit older. And also the UK Biobank, which has 500,000 of people fully biobanked, fully genotyped. And we're just waiting to see those people getting older and see who develops valvular heart disease. Personally, I think there might well be a genetic component in valvular heart disease, especially in the younger population. And we know that with bicuspid aortic valve, there is a genetic predisposition. Somebody who develops aortic stenosis when they're 80, 85, 90, I'm not really convinced that genetics will play a big part. Whether we might be able to adjust the timing of the treatment or the treatment offer based on some genetics, that's a separate situation, and that would be very, very appealing. But at the moment, certainly we don't have anything to suggest that the vast majority of more mature patients that have valve disease is due to genetics.
Thanks. Thank you, Vass, for a really fascinating and impressive talk. As a public health doctor, I was interested in your clinical benefit and the Kaplan-Meier curves you were showing. And so my question is, if you could you give us an idea of the kind of relative scale of benefit of the biomarkers and the MRI scanning on your group of patients compared with kind of readily available population measures like blood pressure, presence of AF, previous stroke, TIA, obesity, those sort of easily obtainable measures. And I have no idea kind of, how the size of risk prediction and benefit, the added value of taking the extra biomarkers and MRI, really.
No, absolutely. I mean, there is no point doing something if it doesn't add anything over and above what we know. So I would like to say an anecdote. And when we all started thinking about this midwall fibrosis pattern and looking at patients who had died, actually, whilst waiting to have surgery, I came across a letter that said, that this was dating back into around 2000, and it said the MRI was normal, there was some non-specific midwall fibrosis, the patient was well, and then he died in his sleep. It was patients like that that made us initially start thinking, what is happening? Why are these patients with midwall fibrosis dying more frequently? So when it comes to purely midwall fibrosis, the ratio that you're looking for is between 2 and 4, depending on the individual patient. So it's quite statistically significant, but also clinically significant. When you start adding biomarkers, you can build on that. But if you're after a number as a public health physician, it's probably going to be something between 4 and 5. Over and above hypertension, over and above atrial fibrillation, over and above smoking, and the usual known parameters.
Question from the chat.
Vass, this question comes from Pranev. On cardiovascular imaging in the future, is AI or machine learning friend or foe? Will the robots take over?
It would be really nice when the robots can take over, because I can have some more time to do other things. Thank you, Pranav. The question is a very valid one. In medicine, everybody talks with AI as if it's a panacea and it was going to take over everything and we'll all become unemployed. Would be nice. It will have a role, and indeed it has started to have a role already. Whereas before, to report an MRI, in other words, just to do the contours and the regions of interest and be able to say this is a ejection fraction, it will probably take about 20 minutes manually doing them. Then it got a little bit faster, it was down to 13 minutes, and now there are AI softwares that can do it in about 10 seconds, and then you spend about a minute to correct anything that the AI has not got correctly. So in that sense, AI will be very instrumental in the future to allow us to process the imaging that we see. It is also very likely that, you know, in 10 years from now, we will not be scanning standard sequences. We will have AI algorithms that, depending on what the sequence finds, they go and image X, Y, Z, and have a more concise, specific to the point MRI imaging. So the short answer is yes, AI will have a big role in the future. Unfortunately, we will still need to be there to look after the AI.
Vass, do you see a time when this technology will tell us when not to do a valve transplant, or valve replacement, or when it will be futile, or it's not going to change the prognosis for a patient if they've you know, got significant fibrosis in their wall?
A very pertinent question, and I remember being grilled at the funding interview with the comment, are you telling us that we should decline offering surgery to these patients because they might not do well? And it's a very big ethical dilemma. I think what we will get exceedingly better is offering intervention at an appropriate time. And by doing that, we will negate the risk of, you know, losing the therapeutic window and therefore coming to what you describe as having patients who are futile to have an operation. If that situation were to arise, I think we just need to be honest with the patients and say that on the evidence that we have, you could have an intervention that will take you out of action for 6 months, and then you might pass away without being able to live any meaningful life. Very difficult situation, but I'm an optimist, so what I hope is that we'll be able to shift that into offering appropriate intervention at a good time.
Vass, as you know, my interest has been in giant cell arteritis. So I was looking at your images and wondering, have you at any point in time found enough resolution or granularity where you can actually view the coronary arteries as opposed to just the myocardium?
Yeah. Oh, no, thank you for that. So I couldn't put all the research I do in 40 minutes. And a lot of work that we had done initially with Gerald Grill, who was at St. Thomas's and now moved to Dallas. We started looking at the coronaries, and we could actually get pretty good resolution. However, you need a dedicated team and you need time. And then one of the projects we did from Norwich, actually, which Michael Frene had led, was back at the Brompton, where we actually optimized the sequence that the Brompton had to visualize coronary arteries, and you can do it. The downside is it takes about 45 minutes, and therefore it still remains in the research arena, because at the same time, CT sort of moved quite forward, and very little radiation, sub 1 millisievert, 5 minutes, you can get the same information. So MRI is not there with CT, but you can do it. I haven't touched on the sort of inflammatory vasculitis group, and I'm glad to see Yannis in the audience, and we are doing some USPO-related MRI imaging, which allows us to see the activity of macrophages, and therefore we can actually see whether any part of the body has active inflammation or not. And interestingly, last week I was speaking to Michael Quayle from Great Ormond Street, who has been reading our papers, and he phoned me up and said, let me know how we can do this in vasculitis, because I want to image the aorta and do exactly the same process. And I thought this is interesting. And I was looking back at our papers, and actually he was citing what we had written down, which was really nice to see. So yes, there is plenty of scope to do MRI research on vasculitis GCA patients.
Okay, and with that clear pointer towards the future and functional MR, I'll bring this to a close. Can I just ask you to thank Vass once again for a fantastic talk?